The challenge of taking a new scientific breakthrough, our understanding of alternative splicing and how that is altered by mutation in disease processes, and then using a new type of drug, an oligonucleotide, to treat patients, is really daunting. So, the first steps is you've got to show that it works when you have these disease cells in a laboratory. And so you show it works in a laboratory. And then you need to go and show it works in an animal model, because you can't treat humans until you understand that there's a really good chance that it'll work and the toxic side effects are not that severe. So you have to show it in an animal model, but you have to design the drug precisely, produce the drug, and use it precisely in an animal model. And then you usually have to go to a second animal model and show that it isn't toxic. And then you have to produce it in large enough quantities with laboratory standards for FDA approval, because you can't go into patients unless you have a very standardized production method. So you produce the same product every time with good quality control so you don't poison someone. And then you have to take that material and get approval from regulatory agencies locally in a hospital to be able to do a clinical trial. And then if you are working multi-state or you want engagement of the FDA with the FDA. And then you have to recruit patients and physicians who, will, for the benefit of that drug, be willing to take and be engaged in early clinical trials. And those patients have to be informed. They have to understand what, they're getting involved in. And they have to see a reason to get involved in it. And you have to treat enough patients so that the variation from person to person and disease from person to person can be sort of averaged out, and you can see a benefit over top all that variation. And that's just a very brief overview of that translational process.